A very good afternoon to all of you, both our audience in the seminar hall and online. So we have gathered here today to celebrate World Microbiome Day. Microbiome, as most of you would already know, are the community of microorganisms which co-live with the host. So World Microbiome Day is observed to raise awareness about the importance of microbiomes in our lives and their impact on our health, society, and the environment. The theme of this year's this year's World Microbiome Day is microbes in food, be it in the probiotics that we get through yogurt or the yummy dosa prepared using a fermented batter. Microbiomes have been a part of our food system for a very long time. There is also continued research in the area of microbial food to produce alternatives to animal products, single-cell protein, and nutraceuticals. So today to celebrate this day, we have organized a symposium. And to start today's program, may I please request our director, Dr. Sagar Singh Gupta, to kindly deliver the welcome address.
So good afternoon, everyone, and welcome to the speakers. And we have a good list of speakers, and I hope that all of you will be interested, and more number of people will come in. Do text your friends so that more people will come in. It will be an interesting 2 hours journey to the world of microbiome. As you know that as indicated by Alpana, that microbiome is a part of life which all of us are having copious numbers in our individual bodies, but it has a huge implication in multiple facets of the work which we do in NIBMG. A lot of thing which is known about NIBMG is about his work on the cancer. And many of you may or may not know that the microbiome niche, which is there in different types of cancers, is a very hot area of research, and that has been coming up very hugely, the interaction between the cancer cells and the microbiome. One of the things which has happened during the process of this discovery is that microbiome has been shown not only to actually modulate the initiation and progression of cancers, but also it has been shown to affect the immune system. That is the way the body actually responds to, you can say with quote and unquote, the foreign cancerous situation. So along with the tumor-associated antigens, certain microbiome-associated antigens are also now recognized on the cancer cells. And that has been also a very recently, that has come as known as oncomicrobiotics. So this is something which is known, and this is something which at some point hopefully our institute will pay more attention to it and bring in synergy to the majority of the work which is done on cancer along with the microbiome research which is going on. Apart from that, of course, there are lots of areas which microbiome traditionally has been very strong on, like the association between the gut microbiome and the different pathological disorders. Certain less worked out areas of microbiome research are the association between ecology and microbiome, and that is something which can be looked up hopefully at some point in future. The human environment and the microbiome axis, human, which is us, the environment, which is of course the surrounding area, and the microbiome axis, is also something which of course has a huge implication when we go forward trying to understand the concept of microbiome. What I meant to say by this is microbiome study should not be just an exercise of knowing new organisms. We should formulate new questions which can lead to understanding of the disease, but multiple number of pathological disease, which the microbiome research should be able to show a light to overcome it. Whether it is ecology, whether it is environment, whether it is cancer, I touched on 3 different aspects. We should be able to do it at some point in NIBMG and with our partners all over the country. For example, the statistical analysis is a huge plus point in NIBMG. And one of the major challenges I understand our microbiome research is about data normalization. The work which you do has to be uniform with the other groups which are working and the different samples which you collect at different points of time. The data normalization is a huge aspect, huge challenge which faces even with cancer biologists, and I think microbiome biologists are no exceptions to this problem, and I think all of you who work on microbiome actually know about this. The work should also proceed towards understanding the patterns of how the co-evaluation has happened between microbiome and also the surroundings. I mean the surroundings are not only the human beings but also the animals also. So in that way, the association graphs may be discovered, which will allow to predict the co-occurrence of the microbiome in different across the species. So these are some of the areas which we should actually think about when we think about microbiome research. Again, microbiome should not be just doing the so-called low-hanging fruits about identification of the microbiome, but answering very solid questions about how to take it to the next level. Finally, I would also like to point out that many of these microbiomes actually are producers of different metabolites. And metabolism and different pathological and physiological conditions go hand to hand. Those who are in the audience from NIBMG had seen a couple of, maybe 7 or 8 days back, Ullas Koltur was here. And you have seen how metabolism has affected different facets of the physiological conditions. Microbiome is no less. Just think of one step before that. They are actually producing the metabolites, which actually is altering the gene expression in many ways, the chromatin remodeling, which allows the gene expression to happen. So, sometime in future, I foresee that maybe isolating the microbiome and doing their whole genome or 16S is not the only way by which you can actually do a metabolite research. So, linking up the microbiome, not by doing its secondary metabolites which it produces, may be a diagnostic way to take the microbiome research forwards. And similarly, we can, some of these microbiomes have ability to actually emit light actually. So a LED device, if it is possible, it can also be a way to actually identify certain types of microbiome in our pathological microbiomes. Or an RFID, the radio frequency, high radio frequency identification signals, which this microbiome may be engineered to do. So these are some of the areas where I think we should think about the next generation of microbiome research, how we can take it forward. Linking up to your favorite areas, broad areas, like cancer biology, for example, I also gave you certain other areas, so that ultimately we come up to a situation where we can link up to the society, link up to the diseases, because all of you know, or may not know, that government is more interested in solving pathological conditions. Government gives out money nowadays more towards projects which actually solve a condition, a disease condition, and not understanding the basic biology. So formulate your research in such a way that these basic biology questions will always be there, but they'll be little hidden between a broader question of translational biology. So this is what is my thought about this microbiome day. Welcome again. have a great 1.5 hours or 2 hours of brainstorming. And I hope that things move forward in this area. Thank you.
Thank you, Dr. Sengupta, for sharing your thoughts on how we can take the microbiome research at NIBMG forward. So now we'll start our symposium. We have speakers, all the speakers who are listed here. To our first speaker today, very happy to have Dr. Abhrajyoti Ghosh with us today. Dr. Ghosh has completed his PhD from University of Calcutta, and after that he moved on to the Max Planck Institute, where he did his postdoc. On returning back to India, he joined Bose Institute as a Ramanujan Fellow in 2014, and now is working as a associate professor at the Bose Institute. His research focuses on microbial stress response and adaptation, and his lab employs a combination of biochemical, microbiological, and genomic techniques to understand the molecular mechanisms of these responses. Without further ado, Dr. Ghosh, please.
I don't think I have. Okay. Thanks for the introduction, and also thanks to the organizers for inviting me to share some of our research in today's meeting. So, I'm working at Bose Institute, as you already know. So, I'm working in a newly formed Department of Biological Sciences, where we have conglomeration of 7 different departments. I'm working on microbial stress adaptation, where we employ model organisms as well as microbial communities to understand some basic questions, and also some questions that are related to environment. So today I'm going to talk about mangrove microbiome that we are working on for last 10 years, and its dynamics in the backdrop of human intervention. So basically, if you look at the distribution of mangroves across the world, they are distributed across all the tropics and subtropics. They are located in the intersection of sea and land, so they are offering a unique environment where you have mixing of salty water and sweet water. It's called brackish environment, and in that environment, the environment is highly productive. So the sediments are normally mostly anoxic and reductive, so they offer fundamental condition where you can study the anoxic microbes and how they perform different functions under anoxic environment. So also they are highly diverse and very rich both in animal and plant species. And so 10 years back, the mangrove that we are working on were not much studied with respect to the microbiology. So we are working on Sundarban, which is the largest mangrove delta of the world, and it is habitat of rare and endangered animals, including famous Royal Bengal tiger, which is one of the only tiger land in the world. It is also World Heritage Site. So what microorganisms are doing in those environments, they are actually contributing towards the biogeochemical cycling that help in recycling and transformation of various nutrients and thus make the mangrove ecosystem more productive. Uh, at this point, I should mention mangrove ecosystems are one of the most productive ecosystems in the world. And then they also contribute in conservation and rehabilitation of mangrove ecosystems, because this is one of the important challenge that we have concerning the climate change that we are facing for last few decades, and also we are going to face a lot in coming decades. And finally, these microbes are also involved in variety of bioremediation processes that actually help us to maintain sustainable ecosystem in those regions. Now, from the perspective of environmental microbiologists, normally people ask three very basic questions. The first question is, who are there? So when you talk about microbes, the first question that comes to our mind, what type of microbes are there? And then the second question that people ask, what are they doing? So if they are present there, what are their functions in those environments? And then finally, how do they survive and interact? Because in an environment, we have so many changes occurring, especially in mangrove ecosystem, we are having lot of let's say, changes that are occurring because of human activities and also because of natural calamities. And because of all these events, it is also important to understand how the resident microbial communities are surviving and interacting among each other. So, while answering these questions, people adopt different type of strategies. To ask the first question, people adopt the strategy is famous for right now. It's called metagenomics, which was coined by Joe Handelsman in 1998. So there are different kinds of metagenomics people do, and we also employ this technique to understand who are there. And then what are they doing? When it comes to what are they doing, people normally go for isolation, characterization, and functional gene diversity, where you can study individual microbes or microbes as a community to understand how they are contributing and to maintain the ecosystem. Finally, when you talk about the survival and interaction among the microbial communities and also the members of different communities, functional genomics play an important role. Now, so today's talk, I will be talking about three different aspects of our study and also studies performed by different research groups. So basically, there are three things I would like to share with you. The first is the good one. So how diverse is the microbial communities and how they are distributed and what are their activities in Sundarbans. So here I will be talking mostly about the microbial structure and function. Then there will be the second segment, I will be talking about the bad and the beauties. In this segment, we will be talking about the effect or impact of climatic and anthropogenic stresses on microbial biodiversity in Sundarban area as a model ecosystem, where we will be talking about shifting microbial dynamics due to pollution, how the pollution actually contribute towards the changes in the microbial communities. And finally, the ugly thing, which is when we are talking about anthropogenic factors, so we need to understand the long-term implication of this anthropogenic impact on mangrove microbiome. And that is where I will be talking about alteration of antibiotic resistance. That is one of the emerging topics right now around the world. So when we come to Sundarban, which is an UNESCO World Heritage Site, it's situated in the northeastern coast of India. And from our studies and many other groups have performed studies where people have shown that there is a clear concentration gradient of anthropogenic factors in this region of mangrove. And while I am talking about anthropogenic factors, I am mostly interested in mentioning that polyaromatic hydrocarbons and different types of heavy metals, potentially toxic elements, are the two major anthropogenic factors that contribute towards alteration and regulation of microbial communities in this UNESCO World Heritage Site. So I will be showing you shortly what are the effects that we have seen. So when we started our work 10 years or more back, actually first our idea was to catalog the microbial diversity, distribution and activities in Sundarban. So basically microbial structure and function. That was the very basic thing when you start any environmental microbiological work or employing genomics. The first thing you always try to know who are there. So we asked these very basic questions. So this is just a table showing the sampling stations. These were changed. So these 3 stations were selected on the basis of their history of anthropogenic loads. So as you can see, Gotkhali, Jharkhali and Shohidnagar, these were the 3 stations. Gotkhali was the most contaminated one, whereas Shohidnagar remains very low or nearly pristine environment where you have less contamination compared to Gotkhali or Jharkhali. So this is just to show you how we pick different types of sampling stations. Now, the way we started, we looked into microbial diversity. So we started with old school concept where people studied the clone library based 16S amplicon sequences by using capillary sequencing platform. And then in the second half, we also looked into amplicon sequencing based assessment using next generation sequencing platform. These were the 2 approaches we took. In the beginning, this is the clone library based studies where this is the phylum distribution which is common and we could see that it's a dominance of proteobacteria in the sediments, which is a typical signature of any mangrove across the world. And we could see gamma proteobacteria remains dominating in the clone library that we studied in our work. And also, we have analyzed the surface sediments. In general, we could see that the surface sediments are generally much more diverse than the subsurface sediments. And also, when we analyze the oxygen concentration on the surface, we could see it's mostly anaerobic in nature. Now, then, when we identify some of the clones, we identify the genus and species of some of the clones, we try to look at the possible function for each of these clones. And we could see we have different types of functions that we could assign to each of these clones or the identity of these bacterial isolates, clones. And we could see that many of them are reported to be involved in hydrocarbon oil degradation, heavy metal resistance, mobilization, extracellular enzyme production, etc., so on and so forth. And interestingly, we could see one unique pattern, that is distribution of phyla in this sampling station that I showed follows a spatial pattern where stations with similar history of anthropogenic influence share similar bacterial abundance or richness, which is kind of very interesting to us at that time, because whenever we see pollution, we could see that the pollution is actually selecting some of the microbial organisms or microbial species in a particular site. And that was the first hint that we got that we have something happening because of pollution. You will see more when I will be showing you some next set of results. Okay, so we also did amplicon sequencing on a 454 platform, which is obsolete right now. Also, we performed spatio-temporal analysis where we analyzed the samples at different times in a year. Actually, we did a 2-year-long analysis. This is just a snapshot of that. And we could see it's a highly diverse microbiome. Again, dominance of proteobacteria, as we have seen from our clone library-based analysis. And finally, surface sediments are anoxic and dominated by delta proteobacteria. Many members of delta proteobacteria actually belong to an anaerobic or anoxic group of organisms. So we identified many of them in the subsurface of the samples that we collected. Now, before I move on, I should mention, so as because I am giving this lecture to NIBMG, so I decided not to go into the functional aspects. I will not show you, we don't have time for that, but we have identified many organisms that are involved in polyaromatic hydrocarbon degradation, also potentially toxic elements, resistant bomylization, halophilic plant growth promoting bacteria, then plastic degrading microbes, biotechnologically important enzyme producing microbes, antimicrobial producing microbe, osmolytes that we are also working on to understand how the microbes actually survive in varying salinity that we have in Sundarban, and finally antibiotic resistant microbes. So today I will be showing you only one slide on this halophilic plant growth promoting bacteria that we identified and we tested at the field level to know how they can contribute. So we isolated two bacterial species, Cucuria flava and Bacillus vietnamensis. These two bacteria are very special. Why? They are resistant to arsenic, and not only that, they can actually show plant growth promotion in high salt concentration, which is very important. Why? Because, for example, in 2006, we had ILA, all of you know, and during ILA, a lot of salty water actually came to the inland and it affected the crop production. So it was essential at some point of time to identify microbial species that can help plant growth under elevated salt concentration. So that was the, let's say, rationale while we started this work. Luckily, we found these two microorganisms are also resistant to different heavy metals, including arsenic. In South 24 Parganas, we have problem with arsenic. And in Sundarban area, we have high load of arsenic in the soil. So we isolated these two strains. They are capable of thriving at high elevated salt concentration as well as they are arsenic resistant. So when we checked their ability to promote plant growth, we could see that they are capable of promoting plant growth in presence of arsenic as well as in high salt concentration. So we further moved on, we further characterized these two organisms. So what we could demonstrated that these two organisms are actually capable of absorbing and accumulating different forms of arsenic on their cell surface, as well as they actually form, when you inoculate to the plant root, they can form a biofilm-like structure on the plant root surface, and thereby they interact with the plant, and following to that we could see they actually contribute towards the growth and development of the plant. Not only that, we also identified or demonstrated that these organisms, when you inoculate to the plant, what happens if you have arsenic in the soil, the arsenic normally goes inside the plant, they get absorbed and accumulated within the plant tissues, different parts of the tissues. Upon inoculation, we could show that the accumulation of arsenic within plants actually goes down drastically when you inoculate with these two organisms, because they absorb and accumulate arsenic on their membrane structure. So that was something that we did. This is just one example that we have performed by culturing some of the microbes from Sundarban. Now moving forward, so now it's from good to the bad and beauties. Why beauties? Because so from the perspective of basic scientists, it is always interesting to understand how human intervention contribute towards changes that is occurring in microbial communities. Although that may not be healthy for either for the microorganism or microbial communities or for us, but that is very interesting scientifically. So what we did here, so basically what happens, because of the settlement, lot of pollutants. Here, the two pollutants I have already mentioned to you, polyaromatic hydrocarbons and potentially toxic elements, they are being released into the natural environment of mangrove. And as I already mentioned to you, our group and also many other groups have shown that there is a clear gradient of these polyaromatic hydrocarbon and potentially toxic elements in the mangrove environment of Sundarban. So we asked two very basic questions. What is the dynamics of mangrove microbial communities in Sundarban? And also, what is the dynamics of functional genes that are present within this pool of microbial communities? So the first hint that we got long back was on archaeal diversity. So basically, here we didn't we chose to go forward with selecting the sampling stations. Here we worked with a like 100-kilometer span of samples. So basically, Gothali is here, and here it is Dhulibashani or Kolosh. It's basically Dhulibashani as it's shown. In between, you have Bonicam. So these are, Bonicam is very famous, touristic place. So now, when we looked at the archaeal diversity in these sampling stations, we could see there is a clear difference. And the difference was like haloarchaea dominates the polluted sites. Whenever you have pollution, more pollution, let's say, with respect to, again, polyaromatic hydrocarbon and potentially toxic element, we do see that we have domination of or more abundance of haloarchaea in those sampling stations, and they are directly correlated to polyaromatic hydrocarbon and potentially toxic elements. And at this point, I should mention, mangroves are normally well populated with methanogens. That is the, let's say, key species in any mangrove environment across the world. Interestingly, we do see methanogens, but the methanogen diversity or methanogens abundance is much high in regions where you have less pollution, which is the typical mangrove environment. So this was the first hint that we got from our study that pollution is actually contributing in a shift in the microbial community, and that was with the archaea. To this end, I should also mention that in bacteria, this was never seen because in bacteria, they are physiologically much more robust, and there is a lot of degeneracy in their physiological function within a community, and that's why we couldn't pinpoint how the shift is going on. I will show you some more results. This is just a statistical correlation. As you can see, these two stations, Bonicamp and Gothali, they are highly correlated with the heavy metals, as you can see here. And also, as you can see here, Halobacteriaceae. So this is basically one of the haloarchaeal species that we identified in our data. So they are the major genera that we identify, and they are all correlated with the Gothali and Bonicamp sampling sites. At the same time, all the polyaromatic hydrocarbons are also correlated well with the Bonicamp and Gothali. So there is a correlation. Whenever you have more of polyaromatic hydrocarbon and potentially toxic element, you do see there is abundance of haloarchaeal species. Okay, so we proposed in this paper that haloarchaea as proxies for hydrocarbon pollution. One of the reviewer was not happy. So he said, well, are these archaeal cells metabolically active? Because when you collect DNA from environment, you can collect DNA from dead cells as well as live cells. So the question remains, whatever you are analyzing, whether that is from the metabolically active cells or they are all dead cells. So to further understand whether they are active cells or not, we collected the total RNA from the soil. We prepare cDNA using 16S amplicon sequence or the primers that we use, and then we perform the same amplicon sequencing from the cDNA. And here also you can see in Gothali, we have lot of Euryarchaeota, means Haloarchaea belongs to Euryarchaeota. So here you can see Halobacteria is dominated in Gothali again, and also halobacteria at the general level, you can see again they are dominant at the RNA level. So we further looked into, this is just a number of reads that we identified in different sampling points, and you can see methanogens are only present in Dhulivasani, these green ones, but the haloarchaeal species are dominating in the Gothali and Bonicam again. So these further actually supported our previous data where we have shown that haloarchaea remains a proxy to pollution. Now, very recently we have moved our analysis from amplicon sequencing to whole genome metagenome analysis. And there we could see few things just the level. Here we have increased the sampling stations. We have included few more. And in this picture, it is much more easy to understand. As you can see, this is much more populated, or rather it's near the inhabited area. So you see it receives much more pollutants compared to this station. So that is why we can always say this is the most polluted one, and also from our analysis, we have shown that. Now, when we looked into the microbiome in general, the diversity, we could see in Gothali we have less diversity, whereas in Polosh, which is this station, is having higher microbial diversity. So we concluded that pollutants lower the alpha diversity of resident microbiome. So whenever you have more pollution, direct thing is some molecules or organisms are being selected, so the total diversity goes down drastically. So when we looked into the dominant genera that we identified, or from our WGS data set, we identified, and we could see the dominant, most dominant ones are all from bacterial genera. And as you can see, Pseudomonas and Streptomyces, they are really dominant in all sampling stations. Now, what happened to archaea? They are not the dominant, but they are there. And we could show previously that archaea are the ones that are more sensitive to pollution. So we also checked within our data who are the dominant archaeal genera. I don't go into the detail. You will find a lot of haloarchaeal species in these samples, and that too, in the polluted ones, we have more abundance of those haloarchaeal count data compared to the less polluted stations or the samples, further correlating with previous two results that we published. And this clearly indicates that archaea remains one of the most sensitive within our data set. Now, as because this was whole genome metagenome data, we could actually look into the functional genes that are also there within our samples. And when we looked into the diversity of functional genes, we could see the functional gene diversity is also less in the contaminated area compared to the less contaminated area. And when we looked into the genes that are being enriched in hyper sites compared to genes that are depleted in low or hyper site or depleted in low parasites, we actually could see that the genes that are enriched in hypersites includes genes that are involved in antibiotic resistance, in heavy metal resistance, polyaromatic hydrocarbon degradation, so on and so forth. So these are the least, and we also looked into the polyaromatic hydrocarbon degraders and fecal contamination indicators that were reported from Sundarban area only, and we looked into their distribution within our dataset, and we could see that while going through this data, we could see PAR degraders are most abundant in contaminated sites, and fecal contamination indicators are mostly abundant in Bonicam, and we concluded that that's because of touristic activity that is going on in the Bonicam area. Okay, so nowadays it is very important you compare your data with the global data. So we decided to compare our data with mangrove data that are available on web or in the database, where we have similar history of anthropogenic load. And I will show you there were three data sets from China, one data set from Kerala, where we have similar history of anthropogenic influences. So when we compared with those data, we could see that we have higher diversity in Sundarban compared to non-Sundarban data, and we have certain genera that are differentially abundant in Sundarban, and many of them belongs to Archaea. Not only that, we also looked into the functional gene diversity, and there we couldn't see much of difference with the global data. However, we could again identify some of the genes that are enriched in Sundarban, and many of them belongs to heavy metal resistant, polyaromatic hydrocarbon degradation, and antibiotic resistance. So the last part, which is the ugly part, where we actually explored the long-term implication of anthropogenic impact on mangrove microbiome. So basically, while doing the previous work, we actually identified a lot of genes that are being enriched in Sundarban dataset, and these genes belong to different categories of antibiotic resistance. So meanwhile, in 2017, Ada Yonat was here in our institute. You know Ada Yonat. She is the first female Nobel laureate from Israel. She was there in our institute when we celebrated 100 years of our institute, and I was lucky to had a discussion with her. And she said, in any natural environment, you should see antibiotic resistance. It's very common, and that has to be very diverse. So from that discussion, we decided, okay, in our data, we could see a lot of antibiotic resistant genes. Let's look into the antibiotic resistant genes and their diversity in details in Sundarban. So why it is relevant? So what happens because of human activities, hospital effluents, animal production, human residues, we release the antibiotic resistant genes in natural environment. And that released antibiotic resistant genes through horizontal gene transfer moves across the microbial communities, and then they, through different activities such as freshwater, leisure activities, and through fruit products, they are come back to us with different pathogenic bacteria, sometimes newly born pathogenic bacteria. I will show you. So we started our work by culturing antibiotic resistant bacteria from Sundarban. And while doing so, we actually identified many bacterial species that are multi-drug resistant, means we tested 4 different antibiotic resistant genes that you can, antibiotics that you can see here. And in this list, these are all resistant to all 4 antibiotics that we tested: tetracycline, kanamycin, vancomycin, and ampicillin. And if you go through this list, you will see many of these bacterial species were never reported as antibiotic resistant from any environment. Not only that, they are typical bacterial species from marine environments. So how these organisms are actually acquiring antibiotic resistance remains a big question. So while doing this, we also decided that because we identified a lot of, like 114 ampicillin resistant bacteria, we decided to look into the abundance of antibiotic resistant genes, which is conferring ampicillin resistance. It's called BLATM in the samples that we analyzed. So we performed real-time PCR by extracting DNA. And when we looked into the abundance of BLATM gene in different samples that we analyzed, we could see in some cases we have huge abundance of BLATM gene. And in some cases we have really little abundance of BLATM. So when we looked into the pollution status of all these samples, we could see the samples where we have higher concentration of polyaromatic hydrocarbon and potentially toxic elements. In those sampling stations, we do see higher abundance of BLATM gene. And we concluded that higher abundance of BLATM gene points towards co-selection of PARs and PTs. This is one of the new theory that is in right now. People who are working on antibiotic resistance are actually believing. So it's called co-selection. Whenever you have a pressure, selection pressure, that selection pressure may not be directly allowing antibiotic resistance dissemination, but still that co-selection somehow allows the organism to adapt in a way to acquire new antibiotic resistant genes. This is one of the new theories, and we could actually propose that in our study. Now, when we completed this study, we also asked two questions. Is this correlation true for all ARGs in the sediment? Whether this is true for not only for Blatim, or it is true for all the antibiotic resistance genes that are present in Sundarban. And second is, how diverse is the antibiotic resistance of Sundarban? So to address this question, we had this WGS data that I showed before. We actually checked the antibiotic resistance gene diversity, and there we could see in the polluted sites, again, we have less diversity compared to the less polluted sites. And when we looked into the abundance, again, the abundance is lower in case of polluted sites. So basically what we could see here, that higher, highly polluted area having less abundance of antibiotic resistant genes compared to less polluted area. And we concluded here that antibiotic resistance diversity is higher in less contaminated sites. Now, we looked into all the antibiotic resistant genes within our data set. We identified more than 30 antibiotic resistant genes, and interestingly, some of the genes are enriched in hypersites, where some of the genes are depleted in hypersites. At this point, I would like to mention, here is the TAME which was identified only in Gothkhali, as you can see here, and also a little bit in Shushnitcha. So in Gothkhali, so this data actually correlates well with our previous data by real-time PCR analysis. So we concluded that no, it's not the case that all antibiotic resistant genes are being influenced by the pollution, but specific ARGs has direct correlation to polyaromatic hydrocarbon. So it's the co-selection hypothesis, and the detailed molecular mechanism is still not known. So again, we compared our data with the global data that is available in the database. Here we could see we have more diversity of antibiotic resistant genes compared to the global data. And when we split it, we could see both Indian samples, Kerala and Sundarban. We could see that these two samples are having more diversity of antibiotic resistant genes compared to the 3 samples that we used for analysis from China. And while we looked into the abundance, we could see we have less abundance of these genes, means the number of these genes within the sample set is less compared to the Chinese data set. And when we perform this cluster analysis, we could see most of them are clustering together. Interestingly, we had a problem that all the Kerala samples were outliers, and also 2 samples from Shenzhen. So initially we thought it was a mistake, or we did something wrong. Finally, after going through the details of these samples, we could identify that all these samples were from the rhizosphere, not from the direct sediment. So in rhizosphere, we have a completely different scenario compared to natural sediment. So we were happy. And with this, I come to our conclusion that, oh, well, because of settlement, we have these pollutants released in mangrove environment, and that mangrove environment actually selectively enriching microbes, and the microbial diversity in general going down, and selectively some of the microbes that can deal with those contaminants are actually now much more abundant compared to others. Now, at the same time, selective enrichment of certain antibiotic resistant genes were also observed. Some of them are also efflux pumps. So we believe that this is because some of these metabolic pathways or the metabolic enzymes that are involved in handling these kind of pollutants use transporters that are also being used by some of the antibiotic resistant genes. So some of the antibiotic resistant process, physiological process, and that's why because of this they have a co-selection, and because of that we do see some of the antibiotic resistant genes are become enriched within these samples. Okay, with this, I would like to acknowledge my lab, the members who are actually doing this work, collaborators, and also the funding agencies, and all of you for your patience. Thank you.
Any questions?
It was a very nice talk. So my question is, you I think probably said that the microbiome that you find in the soil are probably anaerobic.
Yeah, there are signatures of anaerobic microbes, but you see, it's always a mixture. Soil is a very complex. It's not like gut. It's a very complex. So you do have all everything, and sometimes it's very interesting that some of them are in persistent stage. They are live, but they are in persistent stage. As soon as they get proper environment, they start growing.
And is the sampling site covers the area where there is uh, where there are low tides and high tides, or
Yes, they are all. They are all of the sampling sites are like that. So when we collect samples, we have to always throw first 2 centimeters, and then we collect the samples at different depths, and then we analyze. Yeah, and also we have 3 biological replicates, 6 technical replicates. Yeah.
Thank you.
So interesting talk. So, do you think that the process is actually reversible? Suppose a place which is highly touristic and which has a set of microbes, will it be reversible if suppose steps are taken to make it less touristic?
Yes, thank you for this question. So basically, I am currently trying to do some experimental evolution studies where we are trying to actually reverse this process. So we are using some, let's say, in lab, and we are trying to create in lab environment where we have already contaminated soil, and then we are allowing the soil to leach all the pollutants, and then trying to see how the microbiome is changing. So I think this is what we are trying at the moment, and we believe that this might be the case because in another study, we are working on some of the haloarchaeal isolates that are capable of. So in Sundarban, it's very interesting that during summer, the salinity is salinity is quite low because the water level is low. During monsoon season, we have lot of water comes and lot of flow also comes, so salinity goes high. So it goes from 8 to 30. It goes like that. So it's a huge change. But we do see throughout this period, some of the organisms are there, and they are there. So we are now becoming interested to know how they are surviving in this huge range of salinity. While the salinity is changing, also this pollutant concentration is also changing. So that is why we are now interested how at the genome level or at the physiological level, how they are coping up. So maybe, I believe personally, yes, it is possible. The only way is just remove or reduce the regular load of pollutants. That could be one of the.
And a small last query is that in the communities which live in this, there are certain communities still living in this Sundarban also. So have you checked for the prevalence of antibiotic resistance in those human communities?
Myself, not. But I think this is one of the very important questions somebody should address. This is something very interesting. I am also working with some sponge species from Sundarban, and we are so in mangrove river, you cannot collect sponge species because it's a muddy water, so you cannot find sponge. But what we are doing, we are collecting sponge from different ponds in different parts of Sundarban. And while analyzing the microbiome of those sponges, we also identify a lot of antibiotic resistance. So these waters are being used by those people in the villages. So, but it's very interesting to check.
Uh, Great talk. I have a very basic question, which is that you have been assigning all of these changes to anthropogenic reasons.
Yeah.
Now, the gradient through which you have collected the samples and the trend that you observe, there are lots of things which are changing, right?
Yeah.
The salinity of water, the salt content, the oxygen content, etc.,
Yeah.
etc. And as we have mentioned, not to include the dynamics of seasonality, etc. So how do you assign causality to the anthropogenic contents where everything else is also varying here?
Yeah,
May I ask you,
so-
what is your control experiment here,
Yes.
or the
So-
control analysis which
Yes.
eliminates?
Thank you. So in every study that we have performed, we have analyzed all those parameters as well. So we have data for all of them. The variation is mostly, regarding concerning these two pollutants are really variable. What we never measured was the load of antibiotic residues. That we never measured because we don't have the facility to measure antibiotic residues, because nowadays people are also measuring that. That we never measured. But in general, the other parameters, the physicochemical parameters and other different pollutant parameters that we measured, they are changing, but the changes are not significant. These two were consistently significant from the very beginning of our study. That is one thing. And the other thing that is very significantly changing, that is the salinity. With time, we could see that the salinity is playing huge role. But yeah, here I concentrated on the anthropology. Yeah. Yeah. Yeah.
So how do you know that this is some of the place is, or how do you, maybe I'll ask you a more specific question. How do you delineate the selection due to salt versus selection due to this?
Okay. So this is not only us that we are showing for the first time. Around the world, this is now, because whenever you see the changes in the functional gene or the diversity of functional gene, you will see the functional gene diversity includes only heavy metal resistant genes, antibiotic resistant genes, and somehow complex organic molecule degrading genes. So they are always enriched somehow. So that gives us the idea that you can then correlate. Okay, so this is not only us, there are other people also showing the same thing. And also, people are now also, we are doing it. So we have seen that if you induce, as I mentioned, that we are doing experimental evolution studies, if you induce, let's say, heavy metal resistance, okay, if you go on adding heavy metals in a soil system, in a closed system, then what happens, the microbial community is changing. Now you introduce a marker gene, antibiotic resistant genes, you see it's moving in different, through horizontal gene transfer, it is going to different organisms. And those organisms when, no, that doesn't happen. Because in the control experiment, we always have to maintain salt, right? Yeah, that also we are doing. But that doesn't really contribute in shifting of the genes. Okay.
Yes. So, very interesting talk. So, I have a question regarding this antibiotic resistance gene evolution. So, I would expect, I mean, correct me if I'm wrong, I would expect that in an environment where there is high pollution, I would expect that there is more diversity of antibiotic resistance genes compared to, because there is more selection pressure for that.
Yeah.
but you have got the opposite, right?
No, this is the normal. So whenever you are polluting, so if you increase the salt concentration, these organisms love to live in the salt. So there should not be any decrease in microbial diversity or decrease in the abundance of functional genes or decrease in the abundance of antibiotic resistant genes. Now, the point is, in any natural environment, we have a, let's say, a diversity. More or less, we are not going into that. We have a diversity. Now, what you are doing, you are giving a toxin, a pollutant. So what it does, it actually kills a lot of organisms. That actually the reason why we see a decrease in the diversity. While the diversity is going down, you are having less abundance of functional genes, obviously, and also less abundance of antibiotic resistance. In addition to that, what is happening? These toxic molecules is actually selecting some of the organisms. Those organisms now are trying to cope up with the environment, and while coping up, they are also acquiring new antibiotic resistant genes through horizontal gene transfer from the organism residing next to it, and which is antibiotic resistant organism. So that's how this goes.
Okay, so just curious, have you looked at this kind of evolution in your experimental evolution studies? Like,
Yes, we are doing it.
I had a question regarding the arsenic concentration. How did it get into the gradient which you have shown? So you were talking about the trace heavy metals, and you have told that arsenic has also a gradient from have a gradient like the
Okay, so
anthropo.
when I talked about gradient of potentially toxic element, it's not only arsenic, it's overall the concentration of all these potentially toxic elements goes down if you go towards sea. That is normal because all the dumping are on the places where people move around, and what happens, these are dumped as a salt. In many cases, these salts are not soluble in water. So, also this is true for polyaromatic hydrocarbons or any organism. So they are hydrophobic in nature. They tend to get absorbed within the soil particles. That's why you have higher concentration of those molecules where they are being dumped, and there is a little chance that they leach from the soil and goes down and settle somewhere near the sea. So
But
that's the reason. It's not only for arsenic, it's for all the toxic chemicals that we have.
correct me if I'm wrong, but I have also heard that all arsenic can be in groundwater and can also occur in natural water bodies, and it
Yes, you are
leaches
right.
out.
So there are no
So
forms of arsenic. So in natural environment, they try to stay in the insoluble. Okay. So whenever they are being leached, they are leached as the soluble form. There is a transformation event that also depended on microbes in general. And if not microbes, depends on the pH. So if you have certain changes in pH, then the insoluble form can come to the soluble form.
also the gradient of arsenic and other trace element is due to the human intervention or due to the anthropogenic.
They tend to go inside the soil. So that's why and that gradient is not visible in watercolor. If you analyze the watercolor, you see no gradient. Because watercolor is every day you are having a mixing of sweet and salty water. So they are much more dynamic. That's why the microbiome is also not settled in water. You don't see good microbiome in watercolor. But in sediment, the reality, because sediment tells you the history of any environment. Okay, so if you look at the sediment, you see you will have more information. And if you go down in the sediment, especially in Sundarban or these kind of places where some of the sediments are new, some are really good, you can actually study how the microbiome is changing in different layers. So it's really interesting. Also pollutants. you see with changes every year, during every season, you are having changes, but that changes is now becoming drastic. So maybe that will contribute, but more studies are necessary to comment on that.
Well, thank you, Dr. Ghosh, for a wonderful talk and highlighting the importance of microbiome and what we are doing to us, doing to the environment and changing it. So our next talks are going to be from the people from our NIBMG community. So next speaker, may I please invite Dr. Sabyasachi Bhattacharya. So Dr. Sabyasachi has completed his PhD from Ghosh Institute and is now working as a project scientist in the INSACOG project. And he will be talking about, again, environmental microbiome, so microbiome in the Arabian Sea. Please.
Good afternoon, everybody. Thank you for this warm welcome. And my thanks for all the scientists here, all the respected scientists. So today, I'm going to present a small story about our study, what we have done during our PhD time at Ghosh Institute. So, as per our general understanding, we think that, and we have studied in our textbooks, that where this atmosphere, lithosphere, hydrosphere interact within themselves, there forms the biosphere. Now we can propose or we can think it in a different way that microbiome is also a part of this which can control the health of this biosphere. Now, in this way, so my topic is microbiome of the Arabian Sea, oxygen minimum zone sediments, and I will tell a mysterious story about this study. So first of all, we need to know what is oxygen minimum zone. It is generally known as OMZ. So in the western boundaries of the continental slopes, the coastal upwelling push the nutrient-rich water through this way, through this. So the surface water gets fertilized and the primary production increased. Now, as the primary production increased, more and more organic carbon produced, and these get respired in the intermediate layers of the water column. Now, oxygen minimum zone forms when this respiratory oxygen demand exceeds the oxygen availability. Now, what happens? The global warming increases the temperature of the surface water. And this is the place from where the atmosphere and hydrosphere interact, and this is the place where the main oxygen source, that is from the atmosphere, come into the water. Now, as the temperature increases, the solubility of the oxygen decreases. So, and there is also another way to come into oxygen into the water, that is the mixing of the highly oxygenated cold water into the water column. Now, as the temperature increases, the water density also decreases, and this makes the water thermally stratified. So, this lowers the free mixing of the water, and this also make it less ventilated. Now, so the middle layer of the water get low ventilated, and this lead to oxygen expansion. And eutrophication means the nutrient runoff through the rivers and water bodies also influence this oxygen deficiency. Now, we can see the global WMZ position. WMZ can be seen in the Pacific Ocean, Atlantic Ocean, and also in the Indian Ocean, here in the Arabian Sea. And if we see vertically the water column, WMZ zone can be seen as an oxygen deficient water band in between the highly oxygenated water. Now, This Arabian Sea oxygen minimum zone is the most largest and it contains the thickest WMZ and it persists perennially throughout the season. And if we think the total volume of the WMZ water, it is about 10% of the total global ocean. Now, its impacts, like all the aerobic higher organisms like fishes and all other things, they cannot live in this area. And energy flow diverted from the grazing to detritus means microbial metabolism, and it produces methane and nitrous oxide gas. Total 33% means 1/3 of the naturally produced nitrous oxide is produced from the WMZ zone. And methane is also a greenhouse gas, but nitrous oxide is very much potent greenhouse gas. And WMZ is also responsible for 50% oceanic fixed nitrogen loss. Nitrogen is the main limiting source of nutrient in the ocean, and this is get depleted within this 10% zone of the global ocean. 50% is depleted. So it also lowers the primary production. Now, in this regard, we have started our study in the Arabian Sea oxygen minimum zone in the continental slope of the Goa, and we have collected sediment samples throughout the WMZ transect. So this dark color band is the WMZ zone, which touches from the sediment from here to here. This area beyond WMZ zone, this area is oxic water, and again this water is oxic water, and this is the slope. And we have collected samples throughout this slope. Now, here in our study, we have collected 9 such 3-meter long sediment cores, and we have studied each core vertically for about 10 to 15 centimeter interval, means from surface then 10 centimeter means vertically, and each about 3 meter long. And we have done metagenomics, metatranscriptomics, culture-based studies with genomics and geochemistry-based studies for all these sediment cores. And our prime objective was to see the carbon-sulfur biogeochemistry of these sites, and we have seen that these green colored, these sediment cores are present at the center of the WMZ zone expansion. And here, the anaerobic counterparts of these carbon-sulfur cycles were heightened in comparison with the others. So this is already reported. Now, we will concentrate into a small story where we will see that aerobic microbial communities in the sediments of these such sediments, which were already anaerobic processes of the carbon-sulfur cycle was established, and here in all these cores, in the core water there were sulfide detected. Okay, sulfide detected means there is no chance of oxygen. And in this core, this is core number 6, here shallowing of SMTZ was observed. SMTZ means sulfate methane transition zone. So within 3 meter, methane production was started. So this is a very rare event, means 3 meter shallowing of the SMTZ is very rare. And this is only because of higher anaerobic processes was going on. So we were analyzing these metagenomics reads what we have gathered from these sediment points, and we see that there were aerobic respiration related genes by these cytochrome oxidases, and also for aerobic oxidation of methane and ammonia alcohols, along with these aerobic oxidation for thiosulfate, sulfite, and various organosulfur compounds. These type of genes were detected in the metagenomics, and also these were detected in the metatranscriptomics. Then we have checked the culturable diversity. We have done most probable number. This is a tool for checking the live bacterial count in any environmental sample, and we have seen that here obligatory or facultative aerobic chemo-organoheterotroph and sulfur chemolithoautotrophs were very high. Then we have started isolating bacterial strain in pure cultures. And we have selected this sediment point, which is already reported for high sulfide concentration, pore water sulfide concentration, and high anaerobic diversity, and also methane production. And we have selected the bottommost point. Okay. So the water depth of this core was about 580 meter. So just above 580 meter of water, then within the core 3 meter depth. So we have selected that point and we get these 27 isolates. These were categorized in 9 such genus level groups, and we have checked their anaerobic growth and survival along with the fermentative growth or survival. So these are the two options. If a bacteria is growing anaerobically, so they have these two options. And we have checked the anaerobic growth for all the possible anaerobic alternate electron acceptors other than this oxygen. So we have seen that these three, Stenotrophomonas, Halomonas and Pusillimonas, they grow anaerobically. But Gaiparcharia, Methylophaga, Sulfitobacter, another Halomonas, Pseudomonas and Seribacter, these strains could not grow anaerobically or they could not grow fermentatively. So they are obligately aerobic. Now, one clear thing will be like, so they may be contaminants, they may be not actual residuant of that environment. So we have then checked their genomics. So we have checked that they all contain genes for aerobic respiration, and their genetic potential does not have anything for anaerobic respiration, and they also contain sulfur oxidation related genes. And these strains also have complete sulfur oxidation pathway or sulfur oxidation via tetrathionate. And this process also required oxygen, molecular oxygen as the terminal electron acceptor. Then we have checked whether these organisms, so we have isolated these all organisms from one single sediment point. So then we have checked that whether these organisms were only a resident of that point or they were present throughout the sediment course. So we have checked their genetic, we have mapped their genetic genome sequence with all the metagenome sequence available for these sediment cores, these SSK42/5 and 6. That two sediment core which were already reported for anaerobic processes. So we have checked that these within these SSK42/6 throughout all the sediment points they were highly abundant, and also in SSK42/5 they were abundant. Next, we have checked that whether these organisms are only present or they were actively growing. To do that, we have checked the metatranscriptomic data available for this sediment point. We have mapped the genomic data of these organisms with the metatranscriptomic data. And we have seen that aerobic metabolism related genes, genetic information processing, environmental information processing, and also the cell growth and division related genes were already there in the metatranscriptome. So these could be the implications of this biochemical process, like first the oxic ozone in the ocean. So here high primary production, as in the hypoxic zone there will be nothing to degrade these or nothing to eat these high organic matter, high molecular weight organic matter. So these will make organic snow. So this kind of snow is evident in the WMZ zone. So these will deposited, and these sediments will be highly rich in organic carbon. Now anaerobic hydrolysis and fermentation will convert it into simple fatty acid, and sulfate reduction along with all the other anaerobic processes, anaerobic biochemical processes will require this simple fatty acid to convert their products. Now, this is already known. Now, aerobic degradation also can augment the process to produce more simple fatty acid, and also as the members have the ability to sulfur oxidation, they can convert this sulfide again to sulfate, and this will just close the cycle and it will go on. So this is the biogeochemical implication of this aerobic bacteria. Now the most important question will be there, how these aerobic microorganism could survive in such an anoxic zone. So to answer this question, we have checked our metagenome, and we have seen that only there are 2 or 3 such possibilities where oxygen can be produced. So only one such process, mechanism is perchlorate or chlorate reduction. This is such a process where perchlorate ClO4 is converted into molecular oxygen and chlorine, and these genes were present there. We have detected, and also these were presented in the metagenome and also in the metatranscriptome. And we have successfully enriched the microbial consortia with perchlorate. And we got the perchlorate respiring microbial consortia anaerobically, obviously. And we have checked in co-culture experiment, anaerobic co-culture experiment, these can help this Guipercaria strain, which is already known to be obligately aerobic, to grow anaerobically. But in the pore waters of the sediments, no perchlorates were detected. There is no perchlorate in the detected limit. Now we have checked in literature. So we have seen that no such habitats were there where perchlorate reducers and perchlorate both are reported. So this perchlorate reduction process is a such process. It is phylogenetically diverse. Microorganism can do this process. And these type of microorganisms are reported from various environments. But the environments where the perchlorate reducers have been reported, perchlorate cannot be detected. Why? Because these perchlorate reducers can respire oxygen and even nitrate in performance or in the preference over perchlorate. So if oxygen and nitrate is present, they are not using perchlorate reduction. And even and when this oxygen and nitrate is absent, then they will start reducing perchlorate. And in such a way that if then oxygen come into the system, then the perchlorate reduction will irreversibly stop. So in such system that where oxygen is not present, so perchlorate reducer can produce the perchlorate, reduce the perchlorate and produce oxygen. But if that oxygen is just deposited, or there need to be some other organism to use that oxygen. So these aerobic microorganisms may be in association with this perchlorate reducer are using that oxygen, and this makes the oxygen concentration as low as from 2 to 6 mg per liter. This oxygen concentration is required by the perchlorate reducer to reduce or to continue the perchlorate reduction. So this can be the potential for the aerobic microorganism to continue their growth. So this study was done in collaboration with Ghosh Institute and CSIR National Institute of Oceanography, and this was our team, and this was Dr. Ananda Majumdar from NIO. He was our collaborator in this study. And thank you all.
Questions?
It was a nice talk, but my question does not relate to microorganism. My question is why there is a why the hypoxic zone is in sandwich between the two oxic zones.
For this, you need to go to the first. Look here. The source of oxygen, one is from the upper side, another is from the downside. Okay. So here, the atmosphere, hydrosphere boundary, free mixing of air with water, and from here, cold oxygenated water coming from the cold regions of the sea. Now, this middle layer cannot get oxygen from the upside also and from the downside also. That's why it makes a sandwich kind of thing in between the two oxygenated layers, and this is also in the western boundaries only, not in the eastern, nowhere in the eastern boundaries. Because in the sea flow along with the revolution of this earth, these always go into this way. So only OMZ forms in the western boundaries, not in the eastern boundaries.
No more questions. So thank you.
Thank you.
Thank you, Sabyasachi, for your presentation. Now, our next speakers are Mr. Shankarnath, who is, of course, doing his PhD with Dr. Shouvik Mukherjee and Dr. Devjit Dey. They are going to now talk about microbiome in the upper respiratory tract for the SARS-CoV-2 infections. So, over to you.
So, thank you for letting me present. So, I'm going to, this presentation will be very short actually. This is a short but an interesting presentation. So, first of all, I congratulate everybody for the World Microbiome Day. So, today my title of the talk is Upper Respiratory Tract Microbiome Profiles in SARS-CoV-2, Delta and Omicron infected patients. We have survived the pandemic. The pandemic caused by SARS-CoV-2 virus is continuing to be the reason for many deaths globally. The upper respiratory tract, in short, we call it URT, and this URT microbiome shares similarity with the lung microbiome, and it can modulate the host response to the virus. And during the pandemic, there are several SARS-CoV-2 variants that have emerged and gone, and but these several SARS-CoV-2 variants have actually gave differential impact to the lung health and immune dysfunction. So in this image, this has been taken from a Lancet study. It has been showing that throughout the 2021, Delta has been the predominant strain, and from the beginning of 2022, Omicron has actually replaced the Delta variant. But majority of these studies that have been done on URT microbiome and SARS-CoV-2, they have done on case control kind of studies. But no studies have actually investigated the effect of any specific SARS-CoV-2 variant on the dysbiosis on the URT. So our objectives are to investigate the difference in URT microbiome composition and diversity between COVID cases and controls, and further investigate the specific taxa that are associated with Delta and Omicron variant infected patients. So this is our basic study design methodology. We have included 43 COVID patients. During the pandemic time, we could have done this much actually. And we have also tried to collect healthy controls. The sample size is 19. And among the COVID patients, 24 were Delta infected patients and 19 were Omicron infected patients. So the healthy controls, they have not reported COVID-19 for the last 2 years until the day of the sample collection. And all the patients and controls, they have not taken any antibiotic for at least 1 month prior to sample collection. This is very important because 6 months or the microbiome study actually requires that no antibiotic should be there because it will change the microbiome composition. And we have also taken a negative control because this is a study. So negative control should be taken for adjusting the negative contaminants or anything. So this is the demographic. We have taken the patients, controls and healthy controls. We have tried to take them within a single age bracket, like 35, 37, 36. And we have collected samples from nasopharynx and oropharynx. Together we call it URT, upper respiratory tract. Isolated the DNA using Kayam Biostic Bacteremia DNA isolation kit. And all these things has been done under BSL-2 with proper care, proper safety, and we have done 6-ness RNA gene sequencing for the V3, V4 region using NovaSeq 6000. So this is the overview of the sequence data analysis. All the first few files were actually analyzed using KIME2 pipeline. KIME2 is available in the internet, and the basic steps are denoising steps. It requires chimeric sequence to be removed. Quality filtering to be done, and merging of the paired end reads, and also ASV clustering. ASV essentially means amplicon sequence variants. I'm not going to the details of quality filtering and all those things, but ASV are the amplicon sequence variants, and these amplicon sequence variants were actually generated by pairwise comparing 100. If they are 100% sequence similarity, then they are clustered as a ASV. So this has been done using data. Then we have obtained the representative sequences for these ASVs and also the table, ASV table. These representative sequences then were actually gone for taxonomy classification using a reference database that is called Silva 138 database, and the aligned sequences were then also gone for phylogenetic tree development, and phylogenetic tree development could actually give us the alpha and beta diversity estimates. Alpha means the intra-individual diversity, beta means the inter-individual diversity. So this is the result. I'm starting with the results. So what we have done, we have compared the alpha diversity estimates between COVID cases and controls. As you can see, the COVID controls showed very high alpha diversity, intra-individual diversity. There are two statistics like Shannon and Chow, and for both these statistics, we have found that controls are having higher diversity. And we have also compared the COVID cases and controls for the inter-individual diversity, that is called Baker-Tills dissimilarity index, and we are seeing that the controls are actually clustering more close compared to the COVID cases. So it means that based on the diversity, microbial diversity, the control samples are more similar among each other compared to the COVID cases. So then we have tried to identify if any specific taxa that are associated with COVID cases and controls. And so what we have observed that specific taxa like Streptococcus S5MCI, Neisseria, Veillonella, Corynebacterium, Bifidobacterium, those are actually found to be significantly discriminating control samples from the COVID patients. But they are also actually previously reported to be common nasal and oral commensals. So we have found them to be high in control. But in the COVID cases, Stenotrophomonas like Vibrio, Halomonas, Pseudomonas aeruginosa, they were found to be high in the COVID cases. So COVID is like a bad environment actually in the URT. So then what we have done, we have subgrouped our COVID patients based on Delta and Omicron. That is the interesting part. What we have observed that the control individuals, like previous observation, control individuals showed higher diversity compared to other two groups, Delta and Omicron, but Delta has the, compared to other two groups, Delta has the most less severe diversity. When similarly for the beta diversity estimates, like inter-individual diversity estimates, we have seen the control individuals are again clustering together, but controls are, Omicrons and Delta are actually clustering separately. If you closely look it into the plot, you will be able to see that some of the Omicron samples are also actually coming to the control cluster. So it means that some of the Omicron samples microbiome composition is somewhat to be similar to the control samples. So then what we have done, we have again tried to do linear discriminant analysis to find out the specific differences between Delta and Omicron based on the microbiome composition, and observe the similar kind of control bacteria like Streptococcus S5MCI, Staphylococcus caperis, Staphylococcus salivarius, Neisseria, Veillonella, those that are actually previously, other studies have reported that they are common nasal and oral commensals, they are actually high in Omicron, along with some of the non-commensal or pathogenic bacteria like Pseudomonas aeruginosa, Vibrio. So Omicrons are actually is a milieu of both healthy and non-healthy kind of bacteria. But in Delta, they are completely different. They are completely different from Omicron and also from healthy. They are actually enriched with mostly Klebsiella kind of species, Klebsiella pneumoniae, Enterobacter modi, Acinetobacter baumannii. So these species are actually known mostly oral pathogens actually. So then what we have done, we have tried to include the control component here, because we tried to identify the microbiome composition, how can actually they differentiate three groups together. So what we have done principal component analysis based on the microbiome composition. This is separate from the previous ones because previous ones were done using diversity estimates. This analysis, principal component analysis was done using the microbiome composition. And what you have observed that across the PC1, we can actually separate controls and deltas separately. When we introduce a second PC2 component, then we are able to separate the Omicron samples from control and delta. So Omicron is somewhat in between, between the control and delta. So further analysis will be, there are a few slides. Further analysis, we have done some further analysis, and those further analysis has been done by our data analyst Devjit. So he will be presenting the last few slides actually.
Hello everyone. So just Shankar said that based on the microbiome composition, these groups are separated from each other. So now we are trying to understand what are the microbial specific species are there to distinguishing these three groups and try to find out is there any trend present between these three groups. So for that we performed the trend analysis and found these are the 9 bacterial species are the responsible for the differentiating uh, these three groups, and they have a very interesting and significant trend among these three groups. These 9 bacterial key species are mostly abundant in the healthy groups, lesser in the Omicron, and more lesser and significantly less in the Delta group. And they are reported as a commensal for the commensal bacteria found in the URT and mostly abundant in the healthy individuals. And from this analysis, we try to investigate what are the discriminating ability present or having of the microbial community to discriminate these three groups. And for that, we perform the random forest based machine learning prediction analysis, and we observe the microbiome compositions can effectively discriminate and predict the Delta, Omicron and control group with the 90% of accuracy. And here also we've observed that though previously identified 9 key bacterial species contributing the most discrimination of these three groups. So from all of this total study, we finally conclude that the URT microbiome of the Delta Omicron variant infected patients are more similar to the healthy controls compared to the Delta variants. A milieu of commensal and non-commensal species are found to be discriminating for the Omicron variant infected patients from the Delta, but in case of Delta variant infected patients, we found they are mostly non-commensal species. Suppose, for example, Enterobacter mori, Acinetobacter baumannii, Klebsiella pneumonia. So in a nutshell, our study provides evidence that the difference of SARS-CoV-2 variants have the shape, the URT microbiome, which might impact on the overall lung health, and this might implies in the studying the nature of newer variants that might help in the understanding the trajectory of the pandemic. So we would like to acknowledge the Professor Sagar Sengupta, Director of NIBMG, Professor Soumitra Das, ex-Director of NIBMG, Professor Arindam Mitra, Associate Director NIBMG, Professor Pathapati Mojumdar, National Science Chair and Founder Director of NIBMG. And we would also like to thank to JNM Hospital for providing the samples, patient samples, and NGC for the sequencing. Thank you.
Both Shankhoda and Devjitda. That was a really nice presentation. And I really liked the study design and what you have done actually. So you have seen a clear trend between Delta and Omicron. That's right. Overall diversity and also species level also you have checked. Okay, like this is quite interesting. And also what we have like basically what from like general perception is Omicron used to be like kind of infecting the upper respiratory tract, something like that. And Delta was creating more problem in the lung, where the actually gaseous exchange happens and all this. So does like this fact has any like, what are you hypothesizing actually? Like this Omicron and Delta are acting differently or like their infection pattern is actually changing the microbiome? Can you just sum up like? Yes. Mm-hmm. Yeah, and also they have a different infection pattern. Yes, yes.
So that is our main thing. So this URT microbiome or precaution actually we say that URT microbiome actually reciprocates a lot because
Okay.
from URT we actually continuously maintaining an So the microbiome composition, it isn't known that it is established that the microbiome composition in URT will be obviously there will be the intersection of the URT microbiome composition and the non-microbiome composition.
Yes,
will be
yes, yes, that is
So
established.
in that way, actually, we are trying to run samples are actually not easy to do.
That is, yeah, yeah.
So URT is a good reciprocal of the lung.
Hmm.
And the studies are now based on actually doing like this, that if you can know the URT microbiome based on if any specific variant is actually affected,
Mhm.
then that can actually help for the newer variants to tackle it.
Okay.
So because if in specific bacterial signature we found as of the variants, so Omicron we have actually shown that the oils are actually very increased.
Mhm.
But in our study, we have also found that bacterial fauna. It is known as a, there are studies that have shown that it can be actually used as an one spray for a probiotic.
Okay.
It can actually decrease other kinds of bacteria, specifically Pseudomonas species. And we have found bacterial fauna to be high in healthy, Pseudomonas species to be high in good. So these kind of things actually these are further studies need to be done.
Yeah, of course. Like my just simply question is like there is a trend between the Delta and Omicron also. Like Omicron is kind of going towards the healthy. They are having the kind of. So what might be the reason? Like what you are thinking? Like why it might happen?
Delta obviously is a more severe.
Severe, yes.
Upon the COVID, COVID outbreak. So probably so we have not actually done any severity association.
Uh-huh.
But what we have done, probably maybe the severity may be the reason that you have been actually.
Yes.
That may be the reason, but we have also association we have done actually for severity. There's a particular disease called Charleston comorbidity.
Okay, okay.
We have tried to associate with the bacteria, but we have not found any significant result. But we have observed that the CT value is somehow associated with two bacteria. That healthy bacteria Streptococcus, we have found it to be positively associated with CT. It means if the bacterial load is decreasing, it means CT value is increasing. The healthy bacteria is also healthy bacteria. If CT value is increasing, it means the bacterial load is also decreasing.
Viral load is decreasing. Yeah.
And we have observed that the Streptococcus is actually positively correlated with the CT. So these kind
Okay.
of association we have observed. Not for the severity.
Yes.
Will not be able to answer that because we have not found it. But there are other studies, few studies that have shown that COVID severity, not based on strains, just simple severity associated with the microbiome.
Okay. Thank you.
And
This is a like
is done in strain level analysis.
yeah. This is a like quite a good like good insight of the like how these two are different among themselves. Thank you.
So these healthy individuals are vaccinated or not vaccinated?
These healthy individuals were at that time not most. So they were actually most of the very interesting. We have actually fortunately samples in the junction of 2021 samples have been collected from October.
I mean the day of infection. Is there?
So we have collected the samples after the second or first infection. So they usually go to the JNF for testing, and from the testing samples that we have taken out alpha, and then we have so these are the first or second.
So basically, the healthy controls at that time would be maybe people who do not have clinical symptoms.
Yes, they do not have any clinical symptoms. They have not reported anything from the beginning of pandemic, from up to the very time of the circulation.
on, right? Are we on?
क्या करना है? Board of Trustees speaker ही नहीं है.
Thank you, everyone, for joining in, and thanks to all the speakers to begin with for putting up their presentations. Our guest speaker as well, for who has come traveling all the way here. Our IT team, a big shout out to them for helping us organize in the seminar hall and also the online streaming. And of course, our director for his support for this event, and our admin and finance who have helped in organizing things and will help in settling the bills, and Mr. Sujit Ghosh who has helped with organizing the food and everything for you guys. So please go out and have your tea and snacks. Thank you.
And thanks to Arjuna, because in my absence,
उनका